Jaramillo, who is an associate professor at the Universidad Militar Nueva Granada. She earned her PhD from Duke University and was later a postdoc at Harvard University. Her research centers on the phylogenetics of piper, as well as on tropical plant conservation and sustainability. Today, she'll present Piper Evolution and Ecology, a peppery tale from the understory.
Thank you very much, Eric, and thanks for the invitation to come here. Almost 30 years ago, I came for the first time at Smithsonian. I'm just going to try to not get very emotional. So that's where my RTP colleagues back in 1995. So I feel like coming to Smithsonian is coming home. My peppery tale are actually 3 tales. One is about species descriptions. So let's talk about how old botany should really be current botany. My other story is about tropical diversity in the mountains. So just a reminder that the tropics are not flat. And my last tale is a witch tale. That's going to be a surprise. So why am I saying all botany should be current botany? Because where I come from, a lot of species are still not described. And worldwide, according to the IPNI records, in the last 5 years, there's about 2,500 species described every year. And 2,500 species that change their name, new combinations. So the new species may come from a diversity of new explorations, botanists in the herbarium looking at the herbarium sheets more closely, phylogenetic studies. New combinations most probably come from phylogenetic studies and new reorganizing of the groups that they belong to. Let me tell you about I'm here today to tell you what I've been doing the last few years, not a compilation of course we have to step on what we have done, but I'm going to tell you about the current things that we're trying to do. So my first story about a new species comes from what I decided to call since last night, the emerald forest. So I was talking to John Kress last night, and I said, well, I'm working on this beautiful forest, and suddenly I talk about this area. It's actually well known for the emeralds, but not because of the emerald forest, but because of the stone. So you know there are a few emerald mines in the world, and few of those are in the area close by this forest. But the forest is absolutely gorgeous. There are many places where you stand, and as far as your eyes can see in the horizon, there is green mountains. It's absolutely beautiful. So in case you want to buy some beautiful green stones, or you want to go and collect some plants or watch birds, you can contact me and I can help you arrange a trip to go there. So this is a plant we found in the area. The area is called Serranía de las Quinchas. And so we decided to call the species Piper quinchacense. And this is very, very simple to find this as a new species because it has peltate leaves. Of course, my problem is very different from John's. He has 190 species of heliconias. There are over 2,000 species of piper, and there are very few of them that have peltate leaves. So it wasn't very difficult to find that this was a new species. What is interesting about this site, besides the place where it grows, is that this species is, now you have to use this, where is it? Where is my pointer? Oh, here it is. So this is the Chocó region. So the Chocó region, we remember it as in the western part of Colombia. And I kept hearing that it comes down to the Magdalena Valley. And I couldn't believe it until I saw it. So that's where my plant comes from the eastern portion of the Chocó region, one of the richest and most diverse regions in the world. So this plant has some unique features as their pelted leaves, and it has steel roots, which is not very common in piper. When we look at the phylogenetics of where this species sets, it's in one of the large subgenera of piper, but also peltate leaves are not only in that clade. Peltate leaves have not evolved only once, but have evolved many times. So the next question, so here I'm not full of answers, I'm full of questions today. So what the next question would be, and the many questions are, why did this morphology evolve? What are the biophysics behind peltate leaves? Why would they be more common in one subgenus than in the others? Why they do not occur in every subgenera? So there are many questions to be laid out there. My next story is also emotional. So you should remember Al Gentry. And what I remember very well was in 1993, I was sitting in the conference room at Instituto de Ciencias Naturales in Bogotá. And Gary Stiles came in to say that he had very bad news to share with us, that there was a plane crash, Theodore Parker and Alvin Gentry had just died. Two very prominent neotropical biologists were in the same plane and they crashed. Little I knew that 30 years later, I would be studying some plants that Al had collected a few days before the crash, the accident. And I decided to name this species after him. So just before that plane crash, they had done this rapid expedition to the Cordillera del Cóndor. Cordillera del Cóndor is like a 2,000 or 3,000 meter mountain chain in the border of Ecuador and Peru. On the top of the mountain, there are sandstone outcrops. Somehow, it looked like to everyone as tepuy-like mountains. They're not shared geological origin with the tepuys, but they're shared ecological circumstances with the tepuys because they're sandstone, they're outcrops. So there are not many. So pipers like it wet, very wet. And then there are very few species of piper that have gone up in the mountains and where there is little soil. So it wasn't very difficult to identify these species are new, but it had been in the piles for over 30 years. But I also want to share something about Gentry. Gentry was definitely one of the most inspiring botanists that we have seen in the last decades. One of the ideas that was pushed forward by Gentry in his paper over 40 years ago, 1982, on the phytogeography of neotropical plants, was this idea of species swarms. There were the large plastic genera, like piper is in the middle. Very, very large genera with lots of local endemics that he proposed would be a speciation resulting from the uprise of the Andes because this population would get divided. I'm still working on that. I know there are new tools like phylogenomics to better answer those questions, and hopefully we'll keep inspiring, and maybe by doing some more research in that will give more tools for answering the questions in the future. The third species is a piper species. If you've gone to the tropics, more surely you've seen piper species. They're very common in the understory. But one of the things that is not very common either is this globose inflorescence. At least I know in the Neotropics a couple of species that have it. But then last summer, I was in the herbarium and I found these two specimens. So this is, I think I put here the type of Piper nudilimbum. And here is what has been included within it for a very long time, for 100 years. So this is what I'm going to call a new species. If you look at, let's look with botanist eyes. They have elliptic leaves, they have ovate leaves, and it's been misidentified by specialists, people that I admire, Trelis and Ricardo Callejas, who are totally splitters. They see new species in every sample. They could not tell this apart. So I've been trying to convince myself that I am saying something that is correct and they haven't seen before. So if you look at the distribution, there is one of them that is up north in Ecuador and Peru, and the other one comes from Acre and Peru. So there's distinctive geographic distribution. I took samples. I have not seen this species in the wild. Would love to. But I wonder, there is one, genetically they are completely different, okay? But more interesting than that was that in 1928, Trelis published a paper, a short paper, and he's based on a sample that he had seen from a botanist from the US herbarium, saying that this would be a new genus. Later he would come back and say that it wasn't really new. However, now we can say Trelis. Trelis up there in the sky, up there, you were right. It is a different group. So here it is. Here is my, so what it was interesting, it was that back 5 years ago, this plant came out in a place in the phylogeny of Piper that we wouldn't expect. So we kept saying that the sequence was wrong. Now I have 6 sequences, and 6 sequences are not wrong. The morphology of their leaves tell us that they should belong to one group, but their genes is telling us that we need to describe, or we need to revive what Trelis was saying 100 years ago. And that brings me to the classification of Piper. Phylogenetics has really enlightened the classification of Piper. This is a 2000 species problem. In 2008, we published a phylogeny, a smaller phylogeny than this, but a phylogeny that included these groups, 11 groups in the Neotropics. Well, if I color code that phylogeny to what we recognize today as groupings, it's only this group. So let me go back. This group over here is the blue one, okay? Now it's 9 different groupings within it. So we are able now to identify smaller groups, and that will help for everything, for identification, for ecological studies, for evolutionary studies, understanding this group better. And how we have done that? We have done that. We unfortunately have not been able to add many new genes, but we have been able to add a lot more sequences. So this is more than 900 sequences are in this phylogeny. And that is what I think it is important to have more genes, but it's important to have more samples and geographically distributed samples as well. But we could not get to this place without molecular biology, because a lot of those groups, we have seen them as important groups for example, there is a group here that is lianescent pipers. We thought they have evolved multiple times, but surely enough, they're a monophyletic group. So phylogeny is really showing us the way. And the other thing is teamwork. Teamwork, we have put together data from lots of people. Now let me tell you about how diversity is distributed along the mountains. Well, when you live in Bogota and people tell you you're in tropical country and people expect you to be in sneakers and sandals and shorts all the time, you're like, no, there are mountains in the tropics. Well, the data we have was collected by William Trujillo. We have, this is in the southern Colombia, in the Amazonian side of the Andes. We have 2 sites between 500 and 1,100 meters, 3 sites in 1,500 meters or whereabouts, and 3 sites upper than 2,000 meters. In each one of those sites, we sample along lines about 400 meters long, and we divided that into 10 parcels and tried to ID all the species that coexist in the forest. So we're trying to understand how is the diversity distributed along the gradient. And we also use a phylogeny from samples from the area. We have about 90% of the species that grow in the area. From fresh tissue, we have their phylogenies. So what we found with the diversity and how it changes the elevational gradient? Well, it seems like species richness is a good proxy of phylogenetic diversity and functional diversity. So if we take, and the lowlands are more diverse than the uplands, and it is significantly more diverse. And especially species richness is a good proxy, but not quite for some of the others. But we know that in the lowlands, we have a lot more diversity than in the upperlands. I was expecting, because of what I teach and I learn every day, that the mid-mountain will be more diverse than the others, but there is a lowering of the diversity. And if we compare this to other results, and a little bit southern into Ecuador, someone had done something similar, but compared to herbivores, where the diversity of piper just goes down as the mountain goes up. So that's a good result, and it's consistent with what others have found earlier. But we wanted to learn how communities were assembled. And here is where I find it interesting and calling for new research and new questions. So with Diego Salazar in 2016, he studied in La Selva, which is flat. I'm sorry. And what he found was that species that coexisted in little patches were more related to each other, were clustered phylogenetically, but they were over-dispersed chemically. Well, what we find, and that's why I think we should be studying the mountains as well, is that although his pattern will repeat in some places in lower lands, it seems like the species in the upper lands have, as the communities are assembled at random. So the spectrum is a bit different when you go up the mountain. And understanding how phylogenetic, why is this picture and what does it mean for the diversification of piper and the species that coexist in every different site in the mountain would be an interesting project to understand. So all of what we're finding about the mountains is not very, it's fairly, I think it would be fairly new, and it's telling us a different story. It's telling us that we've learned, what we learned from one side need to be tested, and it needs to be tested in the mountains, in the tropics. And the other story is about beta diversity. So when you take the species counts and you calculate the Jacquard index and you try to find how are these species distinct in the different parts of the mountain. So if you only look at species, you see that species in the upper mountain are very different, but above 2,000 meters are very different from below. Only the species names, only if you compare stocks of species. But if you introduce the phylodiversity, the phylogenetic signal into that, you see that the split is very different. So the lower mountain species make a cut to the upper mountain species. So, I mean, the lowland tree against the mountain tree, the mountain forest, while here the lowland forest and the low mountain forest are together and the upper mountain forests are together. So the new questions are, what do mountains mean for the diversification of these groups? And what do the mountains mean for the coexistence of these species? What is different in the mountains? So we understand some more about the diversity along altitudinal gradients. The other thing is that we look at morphology. And although I have to say that I've been very interested in understanding morphology because just the evolution of chemistry, I just can't get my head around. Maybe some can explain me some more. But everyone is talking about chemistry now, about the importance of chemistry for the coexistence of species in piper, in Inga, and even in Rubiaceae. But because I want to be different, then I keep thinking that the morphology and physiology is important. And we still have to look into that. But even if we find that the chemistry is the winner, we still have to tell the story about the physiology of plants, and especially when we go up the mountain. And the other important thing about talking about diversity in the mountains is what could happen with these plants and all what we're finding with climate change. That is a good question, and we'll hold to that. So last is my witch tale. And of course I tell you witch tale because my story is really a disease tale, and it didn't sound really looking forward. A disease tale for closing your talk wouldn't be so colorful. Why am I calling it a witch story? Because I'm going to talk about phytoplasm in natural forests. One of the symptoms of phytoplasm disease is witch broom. So let me tell you some more about phytoplasms. So I have a colleague, I'm a professor at the university, and one of my colleagues studies phytoplasms. That's it. And then I've learned a lot about this. And because you have learned a lot about it, you may be in the forest looking more for it than if you're not. So hopefully after this talk, you'll get out in the forest and email me, Alejandra, I found some phytoplasm symptoms. Okay, this is a tritrophic interaction. It's about the plant, it's a herbivore, the Cicadelidae, the leaf hoppers, and the bacteria. These are obligate parasites because they don't have cell wall. So they have to live in the plant. So the phytoplasms either live in the phloem or in the gut of the insect. Some of their effectors, and that's where this phytoplasm, where the symptoms come, affect transcription factors. So TCPs and MATS transcription factors. Let me show you some of the morphology. They can either downregulate TCPs, and TCPs are this family of teosynthetic branch, if that rings a bell. So when you have this disease, then you lose apical dominance. So you produce a lot of small leaves, fresh tissue for, why not, the herbivore to eat. And the other is these other two effectors affect MATS transcription factors. So that means that MATS transcription factors are flower identity organs. So one of them, one of the symptoms is called virescence. So floral organs will become green, like leafy tissues. And the other symptoms is phylloidy, and that's floral organs will become leaves. So now is my Piper story in nature. So we have detected phytoplasms in Piper, and this is a morphology of a phytoplasm affected Piper plant. So here you can see we have, well, I'm excited because we not only seen it in one site. The first site where we found it was in the forest beside the municipality of Teruel in Huila, but also in the municipality of Otanchi, both along the Magdalena River, but very different sites. And what we see here is the wild type inflorescence, and this is what becomes of it when it's infected. And here is the other species down south. So here we have different, you can have all the inflorescence affected. This is the wild type inflorescence, as geneticists would say. This is what the wild type inflorescence, and this is the one that it's been affected. This fully compromised by the disease, and this partly compromised by the disease. When you look at the structures, this is the healthy bract, and these are the result of the different modified organs. So they're leafy structures. Some of them pretty much look like little stamens with a bit of leafy tissue on the top. But we still have to do the genetic diagnosis. I have to say that I have tried it many times, and on Tuesday when the sequences were out, it seemed like we have sequenced the wrong thing. So unfortunately, I won't have that data coming to you. But there are a lot of questions. So this is the first time someone really talks about phytoplasms in nature, but you've talked with people from the agricultural world, from the horticultural world. This is a disease that people are worried about. USDA has a web page called iPhi classifier, where you can enter your sequence and identify the phytoplasm that you have isolated. So this is something that agricultural people are worried about. This is a real disease, and this is the first. But we are the first time that there is reports in nature of this disease. So the question is how prevalent it is in nature. Are there many species affected or only a few? What is their role in maintaining populations and diversification? But my friends who are interested in the disease said, well, nature is just, the forests are just a recipient of disease that we need to be aware of. But another important thing about this disease is, what is the incidence? People that work on this thing think that climate change is really bringing the disease up. So it seemed like they were just endophytes for a long time, but now with climate change, plants are being more affected. that was my end of my witch story. I hope that I have brought 3 messages to you. One is, let's continue doing naturalism in the 21st century. So in the 21st century, we can keep going out in the field. We should be going. Let's go. I invite you to come. But we have new tools, and when we apply them, we have new things to find out. The other question is, let's be inquisitive and dare to look into new questions. That's not what the establishment wants us to ask. And the other is, let's do the best science and bring it to the public. Because many times, at least in my country, I see that we're bringing it to the public, but we're just bringing refried beans. So, like things that we have done so many times, and we just keep saying the stories. We need to be doing good and best science and bringing those new stories to everyone. They're inspiring. Thanks to everyone that has been on my side. We've been working together for many years, as you can see. And before I'm taken off the stage, I want to talk about some great female tropical botanists. Our Vicky that left us some years ago, and I very, very, very admire these 2 Brazilian botanists. Ariane Peixoto, who is a really leader in Brazilian botany, and Elsie Guimarães. This picture was taken 3 days ago. She's 88 and working in her office every day. So I want to be like her. And the last one is Inés Mejia. She was an adventurer. She went all over the forest. And one of her famous messages was, I don't think there is any place in the world where a woman can't venture alone. So let's keep conquering the tropics. And I don't think I have room for questions, but it's break time.